Chào mừng mọi người đã đến với chương trình webinar của Cộng đồng Hóa học Việt Nam Vietnamese Chemical Association và hội được thành lập với mục đích làm cầu nối giao lưu cho anh chị em người Việt Nam học tập và làm việc trong lĩnh vực khoa học và kỹ thuật liên quan tới ngành hóa từ khắp nơi trên khắp thế giới. Thì hy vọng đây là nơi để mọi người chia sẻ và học tập những chủ đề khoa học cũng như là kinh nghiệm học tập, nghiên cứu và làm việc ở các nền văn hóa khác nhau. Và nguyện vọng lớn hơn đó là qua quá trình chia sẻ này thì anh chị em người Việt của chúng ta có thể giúp đỡ hợp tác để cùng nhau xây dựng nguồn lực và cộng đồng vững mạnh. Thì hội mới thành lập cũng rất là non trẻ, cho nên rất hy vọng được sự tham gia ủng hộ nhiệt tình cũng như là góp ý từ mọi người để hoạt động ngày càng tốt hơn. Thì webinar series là một trong những hoạt động chính của nhóm và sẽ được tổ chức hàng tháng với các khách mời là chuyên gia trong lĩnh vực hóa học. Với webinar hôm nay thì nhóm rất vinh dự giới thiệu khách mời là tiến sĩ Henry Võ, hiện đang là nhà khoa học lâm sàng tại Viện Nghiên cứu Ung thư MD Anderson, viện nghiên cứu về ung thư hàng đầu của Hoa Kỳ. Và trước đó Henry tốt nghiệp tiến sĩ chuyên ngành hóa sinh tại trường đại học Houston. Trong chương trình ngày hôm nay thì Henry sẽ chia sẻ với chúng ta về công việc thú vị của một nhà nghiên cứu lâm sàng cũng như là con đường hướng nghiệp từ nghiên cứu khoa học cơ bản. Mình xin phép nhắc mọi người là xin mute microphone để cho buổi thuyết trình diễn ra được suôn sẻ hơn. Và những câu hỏi xin gửi vào phần chat hoặc là đợi tới cuối buổi thuyết trình. Cảm ơn mọi người và bây giờ là mình sẽ nhường mic lại cho Henry. Và chào Henry, cảm ơn bạn đã dành thời gian để trò chuyện cùng VCA ngày hôm nay.
Xin cảm ơn chị Nhân rất là nhiều. Sau này mình được phép được share screen. Và tại vì mình có sử dụng những cái technical term với lại cái experience thì mình xin được present bằng tiếng Anh ha. So first of all, thank you so much for having me here today, and it is an honor and opportunity that I'm grateful for, and I think this is a very exciting webinar series that's going to be useful to a lot of students as well as young professionals. And so today I'm going to break my talk into 4 or 5 important sections. The first one, I'm going to talk about my background relevant with cancer molecular biology. And then I'm going to discuss my roles as a clinical research scientist with an emphasis on medical oncology. And the next one, I would like to touch upon those transferable skills that can be beneficial to STEM students that going to transfer from basic science to clinical research or to the industry. And I would like to share my own personal tips of networking and how to have a good career roadmap, especially when you are in STEM. And last but not least, I would like to briefly discuss the COVID vaccines, the science behind what's going on with the trials at this point, and some interesting information that available to us at the moment. So first, I would like to talk about cancer. I do have a background and graduate research focused on cancer, and it started with a personal reason because I had family members who suffer from cancer, and I believe that can resonate with a lot of you. And so the question that I was asked a lot of time is that, do we have a cure for cancer, or are big pharmaceutical companies hiding the cure for cancer? So I would like to answer that question is that we do have treatment or anti-cancer therapy available for selective or for specific cancer types. And we do have the therapies also available for specific patients that with a specific subset of cancer. But the challenge here is that cancer is a very very complicated disease, and no individual with cancer would be the same. No cancer, no tumors would be exactly the same, because cancer in nature is made of different biology, different genomic makeup, genomic profile. So therefore, this picture is showing if we have a group of cancer patients and we treat them with the same anti-cancer therapy, they're going to be a subset of patients that are going to respond with benefit. They're going to be a subset of patients who are going to experience substantial toxicity. So the goal um, is that if we can select patients with cancer who are going to most likely respond to a particular treatment that would help us optimize the anti-cancer therapy and to maximize the effect of the drug that can benefit the patients. So this is a picture I would like to use. Imagine one of these grains is what is individual patients, and it's really hard to give everybody to give all the individual patients the same anti-cancer therapy and expect them to benefit from it. What would be better is that if we can select patients and match the patients with a specific treatment that going to benefit the patient with minimal side effects, that would be ideal. And therefore, what is leading us is that we are entering the era of personalized anti-cancer therapy, which means that we're going to treat individual patients with cancer with the most effective or the most potential therapy that the patient most likely going to benefit from it. So how are we going to do it? And we're going to investigate, we're going to look at the cancer biology. And so I promise not going to make it a lecture. So here, this is an important central dogma of biology, where if you are a biology or biochemistry student, you're gonna you are probably going to be familiar with it. So at the DNA level, we have a collection of DNA or collection of genetic materials we call the genome, and then the DNA going to be transcribed into the messenger RNA. And this is important. I would like to emphasize this microRNA right here, because this later going to be part of the COVID-19 vaccine, which is very fascinating. And from here, the messenger RNA is going to be translated into the proteins. And if you have a collection, the whole family of proteins, we call them the proteome, which is the whole collection of individual of biomolecules. And these proteins actually play a very important role in the cells in our human bodies, and they directly mediate or drive different cellular processes, including cell growth, proliferation, and for the case of cancer cells, they also drive cancer cell survival, cancer cell resistance to particular therapy, or cancer cells metastasis. So my research project at the University of Houston, when I was a graduate student, is to look at how these proteins are playing an important role in pancreatic cancer cells, and how together these proteins drive drug response and resistance in those cells. And I look at a particular chemical modification or chemical process in which the proteins undergo post-translational modifications. So as you can see here, a protein is made of residues or amino acids. And those amino acids can be chemically modified with different chemical functional groups. And these functional groups can directly alter the protein function and then directly involve in changing the protein behavior or changing the protein functionality. And my research project was focused on the phosphorylation. So phosphorylation is the process where you take a phospho group and you add it to a protein on specific residue like tyrosine or residue or threonine. And that the addition of a chemical group going to change the protein, going to either activate it, inactivate it, or also mediate the interaction of the protein with another protein partner. And what is important about that is that once those proteins interact with one another upon the phosphorylation, they're going to drive the signaling pathway, which is important chemical signal transduction that mediate or dictate cellular growth, proliferation, metastasis. And if we take that into the context of cancer, signaling pathway mediated by protein phosphorylation going to play an important role in cancer, because if we overactivate this chemical process, it would lead to to cells growing out of control, or it could also help the cells survive under harsh conditions including therapeutic or anti-cancer therapy. So imagine if we're working with a group of cancer cells, if we treat them with a drug, and we also call a drug as an inhibitor. If we treat them with an inhibitor, the cancer cells, what it's going to do naturally is trying to survive. And these proteins are either can be activated or being released in high quantity, or the activity can increase to trying to resist to the drug, trying to help the cancer cells survive. So the question is, if we can track the activity of this protein, we can potentially identify the resistant mechanism, so we can effectively shut down the cancer cells. So my research project as a PhD student was to optimize this microfluidic technology to study cancer biology. And this is a 1 centimeter square microfluidic biochip in which we're going to introduce liquid or solution that contains the proteins isolated from cancer cells. And what it does is that it's going to detect the proteins. It's going to trap the proteins inside the individual reaction chamber. And then we can analyze those proteins, which activity they are doing, or which group of other partners that involve in the cancer cell signaling or the cancer cell processes, so that we can have a better understanding of how cancer cells respond to a particular treatment or targeted therapy. So from there, I like this picture. So I completed my PhD in basic science with a focus on cancer signaling pathway targeted therapy. So to simplify that, my PhD research was focused on how pancreatic cancer cells respond to target therapy, respond to anti-cancer therapy. And the tools that we use is through molecular biology technique to develop our microfluidic technology to look at protein changes. And from there, I developed a very strong interest in clinical research, especially how the basic science is being translated or being turned into a medicine. So I applied to the institution and as a role of clinical study coordinator, and then later I transitioned to the role of a clinical research scientist. And in my current role, we focus on the phase 1 clinical trial, as you can see here. And speaking of clinical trial, there normally consists of 4 phases. The phase 1 clinical trial is where we first testing the safety and the dosage of a new molecule or a new therapy. And the purpose of phase 1 clinical trial is to make sure that the drug we are giving to patients are completely safe. And after a molecule or a drug passes the phase 1, it's going to move to phase 2, which is to evaluate the safety, the dosing, and the efficacy of the molecule. So safety first. The first thing is not to look at whether the drug is effective, but if the drug is safe to be given to the patients. And because the ultimate goal is not only treating patient with an effective therapy, but first, importantly, is not to harm the patients. So after phase 2, the drug is going to enter phase 3, which focuses on safety and efficacy. And as you can see here, the number of participants going to increase. So now we make sure that the drug is safe, is working, and we want to evaluate that on a larger population of patients, because we want to make sure that the drug is working, not only to selected patients, but it's working on that can be given to a larger populations that are going to benefit from it. And after all the endpoints of a phase 3 is met, we're going to the drug or the company that's developed the drug is going to apply for FDA review and FDA approval. And once the drug is approved, it's going to enter the market and can be given to more patients who are needing the therapy. And phase 4 is normally called post-approval monitoring or surveillance. What it means is that after the drug has been approved, it's been out in the market, we continue to monitor for the safety and efficacy of the drug. And there are common cases where after it's been approved by the FDA, it passes the phase 3. There are drugs that would be recalled by the FDA for newly reported toxicity. So it's also a common thing. So the ultimate goal of the all 4 phases of clinical trial is to make sure that the therapy is completely safe, and to evaluate the side effects of the therapy, and to evaluate the efficacy of the therapy. And for my role, because we focus on phase 1 clinical trial, the anti-cancer therapy that we are investigating, we focus on the toxicity profile, and also we could also have secondary endpoints in which the responses are being evaluated or investigated. And going back to this picture, the question here is that if we are treating different cancer patients with the same drug, some patients going to have a response, but some patients going to suffer from the toxicity with no benefit. So how do we select patients for a therapy that's most likely going to benefit them? So there are two strategies that we can do it. The first one is that I just introduced, that has not been widely adopted because of the limitation in technology, is to look at the protein activity, which are direct mediators of drug response in cancer development or cancer drug response. But another strategy to do it is to look at the DNA or the genetic profile of the patients, of the tumors. And thanks to the advancement in technology, now we do have the tool to analyze the DNA from the tumors. And then from there, we're going to track down and identify the key drivers of the cancer, which are the mutations, the genetic mutations that associated with cancer development or most likely going to respond to a particular treatment. So the picture showing here is that we are selecting patients based on their genetic mutation or their genetic profile, and give them a targeted therapy that's going to be most likely beneficial for the patient. And currently we do have different panels of biomarkers or the genetic alterations that have been FDA approved to select treatment for patients. And the goal here is that not to give all patients the same anti-cancer therapy, but to individualize, to personalize treatment for patients so that we can optimize the clinical response and the positive clinical response in these patients. So this is the research interest in part of the projects that I'm working on currently. And another project that I'm working on is that I'm analyzing data and preparing manuscript for publication of a trial involving adoptive T-cell therapy. So this is another very exciting therapy which utilize the T-cells taken from the cancer patient and then either modify them outside of the patient in a test tube and then introduce them back to the patient so that they can fight cancer cells more effectively, or these T-cells can also be expanded in larger population to introduce back to the patient. So the biology behind is that we do have the immune system or the immune cells that attack and track down cancer cells, but for some reason these cancer cells escape these T cells and they avoid being killed by the T cell. So this strategy utilizes a technology where the patient undergo different analysis, different medical procedure where their T cells are isolated and then genetically modified or expanded. And then once being expanded or genetically modified, the T cells become more proactive, that they can track down the cancer cells more effectively to kill them. And this is an exciting technology because it allows for individualized immuno anti-cancer therapy for individual patients. it's unique for the specific genomic profile, or it's unique for the particular genetic mutation within the tumor, so that it can be more specific for selected tumors, because not all cancer or not all tumors are exactly the same. So my role as a clinical research scientist is to help the principal investigator with data analysis, with manuscript preparations, and also we work on protocols that being developed with new therapy, with novel combination of therapies, so that we can uh, help patients with diverse advanced solid tumors. Because the challenge is that with cancer is that they're not just one but multiple diseases, and once they get to advanced or metastatic stages, they become very aggressive, very dangerous. So patients with advanced or metastatic cancer or tumors, they do have very limited treatment options. So we like to not only testing for the safety of the new therapy, but we also aiming for effective or efficacious combination of therapies that are going to benefit patients, to give these patients more options. And from my journey is going from basic science to clinical research has been very blessed, very, I had a great transition. And I would like to discuss, what are the transferable skills that STEM or biosciences students can have going from basic science, because during a PhD, most of the time, students would be involved in a lot of basic science research, but if they want to make a transition to the industry or to more clinical related settings, these are the transferable skills that they can focus on. And to me, it happens quite naturally because if students have been through a PhD training, these skills are essential during the PhD studies already. But for students who are doing the undergraduate degrees or pursuing STEM in the undergraduate studies, these skills are also very essential. So data analysis, I analyze data almost every day, look at clinical data, and then utilize critical thinking skills, which is very important. And then communication is also an important skill to have, because as a scientist or as an employee working with a team, you have to be able to communicate with other team members, to be able to communicate with collaborators or the sponsors. And effective communications in terms of verbal communication or in terms of writing are very important. And another important skill is the writing skills. So writing is very essential, because right now we're shifting to a virtual world. So writing emails and replying to emails are very common, and or even preparing manuscript would require a higher level of advanced writing skills. And developing writing or writing concisely would be also essential. And the next one is time management skills. Transitioning from being a PhD student to a scientist, the time management skills have always been very important, because right now I'm not just working on one research project, one or two research projects. I'm working on multiple projects, working on several manuscripts of protocols. So time management is very important, and being able to prioritize what important, what has to be done first is essential. And last but not least, integrity is crucial, because you're going to be dealing with a lot of data. You're going to be working with other people, so making sure that you uphold the highest integrity in your work ethic, in your data is something that you have always to keep in mind, because once you publish the data or once you say something which is inaccurate to people, it's really hard to go back and change it. It's going to hurt your reputation, and it's going to negatively affect the entire team. So that's something that very important to me. And my tip of getting an internship or a job during my PhD studies or after I graduated is that um, students should be more proactive in terms of networking and socializing. And I totally understand the nature um, of STEM major is that they are very coursework intensive, where the students have to study really hard. They have to spend a lot of time reading and studying and taking exams. But in addition to that, students need to understand that um, not having a high GPA is not enough. They have to actively seek out to other professionals, mentors, and young other professionals, asking them to help to give information about the career or the career or specific positions which these students want to pursue. And what I did was that I talked to my professors. I talked to other young Vietnamese professionals who've been ahead of me, and ask them and show them my interest, my expertise, and ask them if they can help me with the jobs recommendations, or ask them to recommend what important skills that I need if I'm going to a specific field or a specific position. And then from there, by talking to a few people first, you're gonna have the opportunity to expand your network. And then by talking to one or two people, you're going to end up talking with 5 people, 10 people, 20 people. And you always have to keep a positive attitude that you're willing to learn, you're willing to work, and you're willing to contribute to the team. And eventually it's become something that natural to you. And when the opportunity comes, you're ready for it. And especially students in bioscience or natural sciences, it's important to socialize more, to seek out, to have a mentor, to have multiple mentors that going to guide you and willing to advise you on specific career track or career development. And I think that's very crucial that I want to share with you. All right, so the next part is that we're going to discuss the COVID-19 as requested. And I do have a few conflict of interest statements to make. This talk is purely for educational purposes only. The information presented are publicly available. I declare no relevant conflict of interest, and I'm not affiliated with or receiving any funding from the companies who develop the vaccines, and I'm not involved in the vaccine development, clinical trials, or business-related matters. And as we already know, COVID-19 is a devastating disease or infection that's affecting people across the world. And fortunately, we are moving forward with vaccine development. And this is one of the key leaders in the vaccine development of COVID-19 at this point. And the vaccine is being evaluated or undergoing investigation through clinical trial phase 3 at this point. And clinicaltrials.gov is a very reliable source of information where you want to look at drug development that undergoing clinical trials. And from here, what information can you get out of it is that you can look at the company that are running the trials, the sponsor, and you can look at the status of the trial, whether it's ongoing or is completed, and what informations are already being or are reported already. And remember, this picture here, we have different phases of clinical trial, and for the COVID-19 vaccine specifically developed by the company Pfizer and its partner is already close to completion of phase 3. And you can see the next step is to undergo FDA review and approval. And going back to the trials, you can see here the information, the vaccines are being tested, including 2 candidates The first one is the BNT162B1, and the second one is another vaccine that's similar to the first one. And the second one has advanced to phase 3. And they first tested 2 of these, and I'm gonna discuss the differences between the two. But the second one has been showing minimal toxicity with effectiveness or efficacy up to 95% according to the company. And so let's discuss a little bit about the science of COVID-19 or the science of the vaccine. So this is a picture a representation of the COVID-19 structure. As you can see, the virus cell is like a little ball, a spiky ball. And on the surface of the virus, there are multiple proteins. And one of these proteins is the spike glycoprotein. What it does is that when the virus trying to infect the human cells, these spike proteins going to act as a hook so that it can snatch on the human cells and facilitate the entry of the virus into the human cells so that they can proliferate, they can destroy the human cells and cause severe effects. If these viruses would enter the lung of the patient, they're going to grow within the lung cells, and eventually they're going to cause the lung cells to die and cause inflammation, and they shrink out the lung function. So and these spike proteins or glycoprotein play very important role in the infection mechanism of the virus. So what scientists have been doing is that they want to turn the virus weapon to a weapon against it. So scientists have identified the genetic makeup of the virus, and they took the genetic materials that responsible for the making of the spike protein on the surface of the virus. And from there, they designed a messenger RNA that mimic the actual sequence of messenger RNA that code for the spike protein. So what we are doing is that we're trying to train our immune system to recognize the important part of the virus, so that if we are infected with the actual virus, we already have an immunity against it. So the spike protein here, we designed the vaccine using the messenger RNA based on the spike protein. So this company actually packed the messenger RNA encapsulated in a lipid nanoparticle, which is going to be helping with the delivery of the vaccine. And once the vaccine is being injected into the system, the particle is going to reach the human cells or deliver the messenger RNA to the human cells. And then from there, the spike protein that mimic the actual protein on the virus is going to be produced in our human system. And then from there, our immune cells or immune system going to recognize them and become aware of the presence of these glycoprotein or spike protein from the virus. So we develop an immunity to it. So when the actual virus come into contact with our body, we already have this surveillance system. We already have prepare for the virus attack, so so our T cells or our immune cells can effectively attack and shut down the virus. So that's how it works. And there are a few publications have been published recently. As you can see, in September, in August, and in October, that explains in more details about the mechanism of the virus, the effectivity, and importantly, the responses in safety profiles of the vaccine in patients. So going back to the two candidates of vaccines that are available, so the B1 and the B2. So the science of these two vaccines is that the B1 is the one that encodes a secreted trimerized receptor binding domain. So remember the spike or the glycoprotein on the surface of the virus. That's important for the virus to bind into the human cell and to to attract itself to the human cells so that it can enter the human cells and release its viral genetic material. So the first vaccine is to mimic just part of that spike protein, and the second vaccine is going to mimic the genetic material that's going to encode for the entire spike protein. And from clinical studies, they have determined that the the second vaccine, the second candidate, would encode for the entire spike full-length protein is associated with less frequent and less severe systemic reactions. So basically, the second vaccine candidate does have a less severe side effects compared to the first one. That's why the company decided to pursue the second candidate. And as being reported, the common side effects related to COVID vaccine are fatigue, chills, headache, muscle pain, and pain at the injection site, which are very common when we get vaccinated. For example, getting a flu vaccine, we can also experience these side effects. And if you want to look at more into the study design, you can always go to clinicaltrial.gov, and this is the trial identification that you can enter, and it's going to list selective details that that help you understand more about the trial. And for example, here they listed the estimated enrollment number, the number of patients or participants that they expect to be part of the trial, and the allocation is randomized, and they also have the accrual start day and the estimated completion day. So this trial, as you can see here, because they are being tested on so many patients, so they expect to complete it in 2022, which is far from here, but right now, because to the favorable responses in patients, the company is now pushing for a accelerated approval from the FDA through the emergency use authorization request. And this piece of information is taken from the company itself and posted on Friday, which is yesterday. And here, what they reported is that the vaccine efficacy rate is 95%, which is exciting. And this is a phase 3 clinical trial, which is the last trial before FDA approval. And they observed that the patient responded well with less side effects or with common side effects that been reported in other vaccines. And so because it's still being under review by the FDA, we do not know the exact date when the vaccine is going to be approved. So what we can be hopeful is that once or if the vaccine is approved by the FDA, the strategy is that million of doses can be prepared and delivered to the populations by the end of 2021. And thank you very much for for your participation, and I'm happy to take any questions that you may have.
Cảm ơn Henry rất nhiều với bài thuyết trình rất hay về nghiên cứu lâm sàng và các project bạn đang làm việc cũng như các skill set cần thiết cho công việc này. Và đồng thời cái sự hướng dẫn của bạn cho các sinh viên muốn theo cái hướng làm việc này. Và đặc biệt cảm ơn bạn về phần trình bày thêm rất là chi tiết để nhóm hiểu thêm về COVID-19 vaccine hiện nay. Và tiếp theo mình xin phép được chuyển sang phần câu hỏi cho Henry. Và bạn Khánh đến từ Đại học California, Santa Barbara sẽ chủ trì phần hỏi đáp này. Xin mời Khánh.
Hi Henry, I guess I'll be asking the questions in English as well, since your answer will probably be in English as well. So one question from Facebook, from Thủy Tiên Nguyen. She was asking you that when the drugs in the trial show some toxicity, especially when they're not easily metabolized, how do you rescue patients from the effects?
Um, Okay, so rescue patient from the effect. So from my understanding, because I'm not the a medical doctor, so I can just answer this question based on my understanding. So if it depends on the phase one or phase two or phase three of the trial, and especially if for the phase one, if the patient experienced severe toxicity, and so when a trial is designed, the anticipated toxicities are also being considered, and the team have already considered what to expect and have medication prepared for the expected toxicity already. And if the patient experienced toxicity of grade 3 or higher, so grade 1 or 2 are mild, grade 1 is low toxicity, grade 2 is mild, grade 3 or higher would be considered severe, and grade 5 is death. So the most important thing when people investigate a drug in phase 1 clinical trial is that to make sure it's safe. And if a drug is causing severe toxicity, and that toxicity is related to or attributed to the drug, not by other factor, related directly to the protocol therapy. So then the that specific protocol may be terminated or may be put on hold, and then the investigators going to evaluate whether the toxicity is caused by the drug or caused by other factors, or could be this disease itself. And in order to rescue the patient, they typically have a team of medical doctors with different expertises. For example, if the patient's undergoing immunotherapy, they do have a list of anticipated side effects associated with immunotherapy, and then the PI of the protocol may prescribe medication to rescue the patient, or um, getting feedback or getting discussion with other PIs to rescue or to handle with the toxicity with the best response, or with design strategy to minimize the toxicity from the patients. So that's why when designing clinical trial, it's very critical to ensure that the toxicity are well managed before the evaluation of efficacy.
Thanks a lot for the elaborate answer. Before the next question, may I ask that you stop the screen sharing so that
Oh yeah, sure.
yeah, people can see more face to face. And next, if the guest here have some question that you would like to ask Dr. Henry yourself, then you can certainly unmute and turn on the video so that you can ask questions directly to Henry. So the next question that I see from I guess Jun here is that she was saying that thanks Henry for a very great talk. And she has 3 questions actually. The first one, I'll read one by one to you. As PhD programs mostly focus on basic science, what can we do during grad school to prepare ourselves for a clinical researcher position?
Um, Thank you for the question. I think what you can do is that you can participate in a symposium or seminars that involve clinical research. For me, I went to the American Cancer Research Association, which is a large meeting from that involved both clinical research and clinical research involved with the cancer or medical oncology. And that's where you get to talk with people who are doing clinical research, and you can gain more experience with them, to network with them. And also, if you're doing basic science, you can start reading clinical research papers, clinical research articles that relate to your current basic research. And that's going to give you insight of what people are doing with um, after the basic science has been accomplished. So if you're working on a drug molecule at basic science level, what's going to happen next? How that basic science being translated to medicine? And and I highly recommend that you seek out to people that doing clinical research. You can find those people through networking events. You can find those people through talking with your professors, or even going to LinkedIn. You can type in, the title, the position, or you can type in the company that you interested in, and then you would send those professionals, those scientists, or people who are doing clinical research, introducing yourself and your expertise, what you would like to do, and kindly request for an informational interview to have an opportunity to work with, not to talk with them and ask them to provide more information. And I did a lot of informational interviews, and my advice is to reach out to more people, and about 5% of them going to respond to you. Those people are medical doctors that I met at conferences. Those are senior scientists at well-established pharmaceutical companies, and do not expect them to give you a job or to give you a referral or anything. Just talk to them with a passion for career development, for personal improvement, for your preparedness of your career. And if after talking to so many people, you're gonna gain more experience of what you need to do. And the next step is that trying to look at companies in your area or in your states that open for internship, that willing to take graduate student or undergraduate student for like a summer 3-month internship. And that is incredibly helpful, because that's exposed you to the industry or the work environment outside of a research lab, and giving you the opportunity to gain more experience and to polish, to improve your resume with work experience rather than just research. And when you're ready to apply, you already have experience. You already know what you should be doing or what you like to do, and that's going to help tremendously in terms of finding a job, or of determining your career.
Thanks a lot, Henry. And the next question is that are clinical trials usually in collaborations with the industry? And how is your work at the MD Anderson's compared to the counterparts from the industry, company, or sponsor side?
Oh, this is a very interesting question. Yeah, so my job actually as a clinical scientist is the institution that I'm working at right now is actually an academia. But I do work, yeah, it's true that we work with sponsor or pharmaceutical collaborators. My job is, how is it different from a scientist in the industry? And this is interesting because before joining as a clinical research scientist, I also consider being a scientist in the industry. So there are different position of scientists in the industry, right? You can be a scientist that work in the lab or overseeing a research project in the pharmaceutical company or biotech company. And excuse me. And being a scientist in the industrial setting, you still have to conduct experiments when you first start. And then later on, you would be able to move to higher position where you get to manage or to oversee projects. But ultimately, it's involved in drug development or study design. And then for my position as a clinical research, working with collaborator, is that I don't have to do experiments anymore, but I do have to be involved in a lot of data analysis and communication. So I facilitate communication between, my team or the PI with industrial or industry sponsors, and help the PI design protocols. And we work with the sponsor in terms of information, the design, and also the data, which is very important. We also make sure that the data is accurate, is well presented, is well organized. So I would say that for the industry, because a truck is like the main product of the company, right? So it's depend on the pipeline as well. When you enter a company as an industrial scientist, are you going to be involved in the department where early development, early truck discovery? So that as that role, you're going to be doing really similar stuff as basic science, right? Because it's early development, you have to test the activity of molecules and other stuff. But if you move further to the later stage, you can be involved in overseeing animal study or preclinical study that's close to or that prepares the drug for clinical trial, and that still require some basic science training. That still require management, and for me is right now, as I feel like is for my job is involved in more of data analysis rather than um, um, experiments or wet lab. Mm-hmm.
Thanks, Henry. The next question, we have another one from Facebook. This person was asking, what skills do medical students or doctors need to be part of the clinical trial team?
This is great. This is a good question. I I would say that as I do not have a lot of experience because myself as a scientist, a PhD background, so I cannot speak on behalf of the medical students who have a different training or educations. But what I think is that from my experience working with my PI is that um, you have to be experienced in medicine first, and you have to have the highest quality of patient care and work ethics and integrity. And for and normally from my interaction with other faculties who are principal investigators for clinical trials, that they have a lot of experience before joining clinical trial department. So they have been treating patients, and they have been involved in specific type of therapies already, and then they were recruited or they were interested in doing clinical trials, and then they would transition to a clinical trial um, um, medical doctor, and not so it's more like a academic setting where the medical doctor have to not just treat patient but to conduct research at the same time. So having the ability to be both a scientist and a medical doctor who are going to take care of patients, I believe that's the most important thing. And you also going to, from my observation, is that you're also going to be involved in a lot of writing, not just simply treating patients, but a lot of writing and management. You have to manage. You not just take care of patient, but you have to manage the entire team that's running the trial or running different clinical trials at the same time. So you not only being a medical doctor, you being a leader, a manager, and a and scientist.
Thanks, Henry. We have another question here, also from Facebook. So the topic is going to change a little now to the COVID-19 vaccine. This person is asking, when the COVID-19 reaches vaccine trials reaches the final phase and is approved, then which safety factors one may still consider to make the decisions to take it?
With safety
So
factor.
essentially, this person is asking that once the vaccination is approved, are there still still concerns, well, so that the consumers would decide to take it or not?
To be concerned. Yeah, this is an interesting question. So once a drug or a vaccine approved, that means that all the safety profile that been reported or analyzed have been reviewed by the FDA. So they should be listed and be available to the patients or to the public. And the source of information um, can be obtained is from the FDA indications, and it would list all the reported or the expected side effects or adverse events there. And as a patient or a healthy individual who consider taking the vaccine, I would go to I would go to the FDA indications, because in order for the FDA to approve the vaccine, they first have to make sure that all these side effects have been reviewed, have been reported, have been analyzed, and if they occur in the patient, what would be the medication or what would be the strategy to manage the toxicity. So make sure that it's safe, and we do have a clear strategy to manage the toxicity before the FDA approved. And once it's approved, it's pretty much that we already know what's going to happen, or what side effects most likely to occur, and we already have prepared for the vaccine. So
Right.
I would be more considerate about the severe toxicity that reported, but but if but if a drug is too severely toxic, it wouldn't be approved by the FDA, because
Right, but then so I guess another question that's related is that you were saying that the phase 4 post approval surveillance is still necessary. Does that mean that there have been cases where the results from phase 4 are significantly different from those from phase 3, such that the drugs or the vaccines have to be basically stopped from being used and whatnot?
Yeah, so I can't think of an example right now, but there are few cases where a drug passed the phase 3 and was released to the market, and then later a severe side effects were reported, and then the FDA recall, or it could be something related to the ingredients in the drug, in making the drug, that it's not the drug itself, but other ingredients in the pill could be associated with some side effects that have not been reported before, have not been observed before, and at that time, the regulatory entity is going to put a hold on it or recall it until that has been completely addressed. So for vaccine, so that's why we don't want to rush or cut corners, because to develop a drug or a vaccine would take a long time, and not because we don't have a vaccine, but that because we need to make sure that the vaccine does what it's supposed to do, and the vaccine is completely safe first and is effective. So after phase 3, typically all these safety profiles have been reviewed and approved by the FDA, and all the strategy to manage those side effects have been determined. And the phase 4 now phase 4 is post-market. So a lot of people are going to be treated, a lot of people are going to receive the vaccines, but they need to make sure that the drug is safe for, even after it's been approved, is it safe for majority of people, majority of patients. So that information needs to be collected.
Right. There's another question from
Can
I
I
think.
have a follow-up question on that?
Right.
Yeah, Henry, thank you for that explanation. I was wondering how the long-term effects of a therapy or vaccine that under clinical trial get evaluated. What is the amount of timeline that we allow to know whether or not there are any long-term effects of a type of treatment that we're testing?
It depends on the protocol drug, and it depends on the therapy. For example, for immunotherapy for patients with cancer, the because the drug modulate the immune system, so it may have delayed toxicity that not going to occur or at the moment when the patient's undergoing trial, but it may occur later on. So that's considered delayed toxicity. And in order to do that for patients who are undergoing novel immunotherapy or T-cell therapy, the FDA would have guidelines that require the patients to be monitored for up to 10, 15 years for side effects. So even for vaccines, I don't have the exact number of years now, but the FDA always have guidelines that if it's interfere with even gene therapy that interfere with our genetics or immune related therapies such as vaccine or immunotherapy, those have to be monitored for long. From my understanding, those will be monitored for a longer period of time, can be up to certain number of years, because we don't fully know what they're going to do to our immune system, because the immune system may or may not respond when we first receive the vaccine, but it's the delay toxicity may occur a couple years later, a few years later. It's accumulated over time, or the immune system slowly respond over time, and that has to be reported. And so therefore the guidelines will be established, and with it typically will say that the monitoring or patient monitoring is up to some number of years, 10 years, 15 years.
Awesome, thank you for that clarification.
Yeah, Henry, we have another question here. What type of data analysis does the clinical research scientist conduct? Is it patient to patient, or is it based on statistics of a large group of patients?
So it depends on the size of trials as well. So for phase 1 clinical trial, as you can see that we're evaluating the safety of the therapy. So so the phase 1 trial is typically working with small population of patients up to 30, 35. And because of that, for my analysis, I would evaluate or would analyze information by individual patients. And then I also conduct statistical analysis on the population of the patients being treated. For example, if the trial enroll and treated 20 patients, then all 20 patients have to be evaluable for responses or evaluable for toxicity. And then when we prepare the manuscript for publication, we can report toxicity by patients, and we also report the common toxicity that occur in a higher population or higher portion of those 20 patients. So being clear, being detailed and transparent is very important. So we have to analyze the data very carefully, very thoroughly, and we have to consider the severity of these side effects, or for me, I also analyze the responses, how long the responses last, how long the patient experience the partial response or stable disease, how long they until the point that their disease progress. And then we have to conduct a survival curve where we identify the time when the patient treated to the point that the disease progressed. This is called the progression-free survival analysis. And overall survival analysis is where the patient, based on the patient was treated until the point that that they are taken off the clinical trial, whether they are survived or that the patient passed away. That's sometimes that's also reported.
Thanks, Henry. We have another questions right here that's probably a lot more relevant to the topic that we're having today. So I guess you've been working in the clinical trial setting for about a year now. What is the main difference that you notice between research in the clinical setting and research in the basic science lab?
The main difference right now, for sure, is that a clinical research scientist do not have to run experiments. Uh, so I used to run a lot of experiments, doing a lot of cell culture, protein purification, isolation, Western blotting, using a lot of molecular techniques. Um, and for now, I don't do those anymore, because remember, for basic science, you work with molecules, you work with cells, you work with animals. But clinical research, we work with patients. We are helping and treating the patients. So, it's like a treatment of patients. It's, in another term, it's like running a larger experiment, but at the same time we're also treating patients. So I don't, because I'm not a medical doctor, I don't treat patient, and it's different from treating cells in the petri dish, right? And that's why, in order to conduct a clinical trial, is required tremendous effort of a team, not just one scientist or one PI. So we would have the PI, who is a medical doctor. We have a clinical study coordinator that coordinates or help with the patient care as well. We have clinical pharmacists that prescribe the drug, and we have data coordinator that's responsible for data collection and data entry. And we also have me, clinical scientist, who doing the work behind the scene. To when the trial is completed, I will help analyze data that's been collected by the team, by the data coordinator, and then analyze and then prepare manuscript based on the information associated with toxicity or responses that been collected or reported.
So it sounds like you're the bridge between basic science and medical science, medical research, essentially. Is that a good description?
Um, I it's it's different in term of daily work, but it's similar in term of analyses. So, as basic science, what you do is you collect data, right? You collect data from your experiments. I I don't have to collect data from the patients, because now we're working with humans, we're working with patients. So the data collection is being conducted by the data team, which is important because we don't want to overlap responsibility, because that eventually going to create non-transparency, or we want to have different people within the same group with different responsibilities. Together, we make sure that we collect the accurate information, and we organize them, we analyze them correctly and carefully. So, so for, I would say, as a PhD student or a basic scientist, if you're not a PI, you a postdoc or graduate student, you do a lot of work from the beginning to the end. You design your experiment, you run your experiment, you collect data, you do analysis, and you prepare manuscript for publication, right? For a clinical trial setting, we break those into different sections, and we have different teams that responsible for a particular part of that trial. We have the PI who's the oversees the entire trial, responsible for patient care or patient treatment, and who also happen to be the most experienced person, of course, right? And so for the data collection, we have study coordinator, we have data coordinator that collect data. If I'm the basic scientist, I would collect data from my experiment, right? But now the PI treats the patient, and then the data is collected by the coordinators, and I would analyze the data. So for basic science, I am analyzing data. Now I am analyzing data, but I'm not collecting data. Yeah. So it's similar if you put them from data, from experiments being conducted to data collection, data analysis, and publication is the process similar. But under the clinical trial setting, we have specific teams that specialize for specific part of the entire experiment, which is the clinical trial on humans. So clinical trial is the a larger experiment on patients, right? It's still an experiment, but because we're working with humans or patients' lives, we have to be extremely careful. That's why we need multiple people to be involved, multiple people with specialized expertise and responsibilities to make sure that the entire experiment or the entire clinical trial is carried out successfully and carefully, so that we can make sure the patients are safe. That's the number one, and make sure that we deliver the best patient care, and potentially, hopefully, this novel therapy is going to benefit the patients.
Right. Thank you, Henry. I guess we just have one last question before we close up this Q&A session. So if COVID-19 vaccines are to be distributed shortly, can we loosen the caution or restriction around COVID-19, given that Thanksgiving and Christmas are coming? Do you have any comments or advice on that?
Once again, I'm not an expert on vaccination or what's the term? But I think I even though we are very excited, very hopeful that the vaccine going to be here, but I think that having it before Thanksgiving or before Christmas would be a little overly ambitious. And as you can see that now is going to be going under FDA review. And we don't know how long it's going to take for the FDA to approve the vaccine, even though the result is very favorable. But it will take at least a couple more months for the vaccine to be approved, and then we have to wait for the company to produce the vaccines and then distribute it to the market, to people. And they probably also have prioritized, right? They have which specific groups going to receive the vaccines first. I would think that the frontline or medical worker going to receive them first, and then how it's going to be distributed to more people. With channel, how it's going to be distributed, and that one critical thing is that because it is a messenger, it's an RNA vaccine, and they have to make sure that it's being stored correctly, and make sure that the vaccine is kept under specific condition where it's being injected, the patient is still working, right? We don't want to leave that at room temperature and then the vaccine will be degraded.
So yeah, I guess because the vaccines are to be kept at extremely cold temperature, so I guess the logistic and distribution of that vaccine would be one hell of a nightmare. If not to say that it's gonna be very difficult and challenging. It's like, I think it's unprecedented because we haven't had to vaccinate this many people within such a short time frame, and dealing with such a challenging vaccine, essentially.
Mm-hmm. Yeah, and and it's not just the vaccine, the science, the medicine, right? Sometimes it has to do with the regulations, the politics. So it's really hard to say, when we're gonna get back to normal, when the vaccine gonna be available to all of us, or how long does it take? It's really hard because there are a lot of factors that we may not be fully understand. That's what's going on behind the scenes, and but we can be hopeful that the vaccine is going to be available by the end of 2021, which sounds like it's really far away, but we already we already close to the end of 2020, so
Right.
you know, a couple more months wouldn't be so much different.
Yeah, we can't wait for 2020 to be over. So yeah, so I think that's the last question of the Q&A session, Henry, and I think I'll leave the narration to chị Nhân now. Thank you.
Xin chân thành cảm ơn tất cả mọi người đã tham gia chương trình ngày hôm nay, và đặc biệt cảm ơn tiến sĩ Henry Võ. Và mình cũng mong mọi người tiếp tục ủng hộ và đóng góp nhóm để ngày càng phát triển vững mạnh hơn. Thì nhóm sẽ gửi một feedback form để nhận sự góp ý cải thiện cho những buổi webinar tiếp theo, và cũng post video của buổi webinar hôm nay lên YouTube để mọi người có thể xem lại dễ dàng. Và chúc mọi người cuối tuần vui vẻ. Enjoy your weekend.
Cảm ơn mọi người.
Cảm ơn Henry rất là nhiều.
Have a great weekend.
Cảm ơn Henry.
Yeah, have a great weekend, everyone.